Karolina. I'm Scientific Project Manager at VUGENE, and at VUGENE we are specializing in multi-omics data analysis and interpretation to support drug development, discovery, with an aim to give a better understanding of mechanism of action, do it faster, and de-risk the drug development and discovery process. So my personal background is molecular biology. From the professional side, I was involved in various organizations, bigger and smaller, mainly on product management and business development side. And at VUGENE, my main role and responsibility is building new partnerships and creating new connections. And definitely, there are a couple of reasons for that. So first of all, speaking about the genomics, historically, it was the first omic discipline to achieve the large-scale standardization that led to dropping in costs dramatically, having matured analytical pipelines quite early on, and just having a large genomic databases become available quite early on. So naturally, the industry focused on extracting as much value as possible from DNA-based information. However, as you mentioned, biology is dynamic, and DNA really provides a good basis. It's a good blueprint, but it doesn't fully explain how the cells interact, how disease develop, how disease evolve over time, and how the treatment works once injected. So to answer those questions, researchers really needed multiple layers of biological information, and to include more dynamic omics, such as proteomics and metabolomics, that really reflect the current state of the system of the body and the organism. So when it comes to multi-omics, the challenge was integrating the omics with data being produced by using very different technologies and different scales, resolutions, and quality levels. So still looking at individual omics such as genomics, proteomics, metabolomics was easier than integrating the omics. But we are now really reaching the point where technology, both that generates the data and the computational power, analytical methodologies, have evolved really sufficiently to make true multi-omics integration possible and applicable in R&D setting. So yeah, at the same time, we see that with the technology evolving, we see how complex disease are, such as cancer, neurodegeneration, autoimmune disorders. So there's really no single layer analysis that can really capture the underlying biology. So I would say that now multi-omics is really coming and emerging as the answer to a lot of research questions. And it was not fully recognized before because we just didn't have the technology, both from the data generation point, as well as the computational analysis to really utilize the technologies fully. First of all, looking from the technological point of view, data fragmentation still remains an issue. We see that different organizations and sometimes different departments within the same organization are generating data independently and are using different platforms, different analytical standards, different storage systems. And as a result, data sets may exist within the same organization, but remain very disconnected with one another. Yeah, so second thing might be generating data has become relatively straightforward, but doing the analysis and interpreting the data really remains one of the challenges. So omics data sets are often collected across different cohorts and experimental conditions, and that makes extracting biologically meaningful signals more complex, and that really requires quite specific expertise. And then, so I would say those are the two technological challenges that come to my mind. And then culturally, we still see some disconnection between wet lab researchers, scientists generating the data, and then computational teams analyzing that. So really bridging the gap and collaborating within the organizations, within these departments is the key. And we see that from our personal experience, that collaboration between wet lab scientists and bioinformaticians, computational biologists is one of the keys. So my perspective is really that the future and the challenges don't lie in generating the data anymore, but it's more about creating the analytical frameworks and connecting diverse data sets that can be translated to biologically interpretable insights and really lead to new discoveries and developments. Yeah, so that's a great question, and it's really relevant in the industry currently. So we see that single cell and spatial technologies really provide powerful insights, but distinguishing real biology from noise remains a challenge. And the industry is addressing that through simply developing more advanced statistical methods and methods for analyzing the data sets, and also applying AI-based foundational models. So we have this model called single cell GPT at the moment that can really validate rare cell populations against large reference data sets. So it's great to see that we utilize artificial intelligence in this way. And also we have more precise denoising tools being developed. One example could be the space bender that is very great in removing the background noise, background RNA noise without losing the genuine signal. So I think when it comes to rare cell types, the focus today is not only on finding and identifying the rare cells, but to ensure that the findings are reliable, reproducible, and really biologically relevant. So I think here we once again come to the interpretation part and just really understanding the biological relevance and meaning. We are very excited about Bio in San Diego this year and the opportunity to connect with the broader ecosystem, as well as meeting our existing clients and partners, and just discussing how data-driven approaches are reshaping drug discovery and development. So we are really, really excited about that, meeting with the industry and having those meaningful conversations. I myself will be there, as well as our Chief Scientific Officer and Chief Executive Officer. And we will have a booth at the Lithuanian Pavilion, and the number is 5143.